Hello everyone, my name is Aysulou Smagulova. We're here at the Viruses 2024 conference organized by MDPI, and I'm joined here with Prof.Dr. Luis Enjuanes. Thank you so much for being here. It's an honor to interview you.
My pleasure.
I will just start with my questions. So, given your extensive experience in coronavirus research, what are the most significant advancements or breakthroughs you've witnessed in understanding of coronavirus replication, transcription, and virus host interactions over the course of your extensive career?
Well, there have been many advances in many directions, but probably one of the most important ones has been that in biochemistry, the DNA molecule, which is very important, during replication has a safety system. Whenever a nucleotide is introduced by mistake in the chain, there is a system that will remove this nucleotide and allow another chance to repair the error. So this is what we call the proof system, proof reading system. It was discovered a long time ago in the DNA well. But now with coronaviruses, for the first time in life, has been also described the proof reading system in the RNA well. And that happened for the first time in the coronavirus well. This is very important because it's a way to repair the sequences of coronavirus. And quite probably happened that because the genome of a coronavirus is 30,000 nucleotides. It's the largest genome known in the world for an RNA coronavirus. So the virus was introducing too many errors in his life.
Right.
And he needed a proof reading system, and it was discovered in this family of viruses.
Wow.
That was very important because also has many relevant implications for the life of a coronavirus.
Yeah, I see. Thank you so much. Okay. As a prominent figure in virology, you've seen the field evolve significantly. How do you envision the future of research on emerging viruses, and what key areas do you believe should be prioritized to enhance our preparedness for future pandemics?
Well, what we have seen with this pandemic now, due to the SARS coronavirus 2, you could predict that will be happening, because historically that happened before. Not as hard as this one, although the flu of 1918 was also a very important one. But people is not aware that every year we have a new coronavirus. So for the last 10 years, we have discovered 10 new coronaviruses affecting humans and animals. So this is the standard in life. So certainly that is telling us that we have to be prepared, not only for coronaviruses, but also for new flu viruses or any other potential virus that can emerge. Today, with all the traveling that people is doing, that we go anywhere in the world without being too cautious, we can guarantee that there will be new viruses emerging. And how to counteract this? All we have to do is in key places in the world, having detection laboratories, highly advanced, geographically speaking, and with good technology, and having very good connection among them throughout the world. So this is essential. These links to prevent the spreading of new viruses already exist, but we have to reinforce them. And reinforce the communication. So sometimes there have been people complaining about whether the sequence of the new virus was released to all the scientists in the world as soon as possible or not. Sometimes there have been a delay, but in general, people has been quite generous because we need the sequence to develop the detection system and to prepare the vaccines. So improving the initial detection and improving the communication is a must for all of us in working in science and for all the diagnostic laboratories in the world.
I understand, that makes sense. Having served as Fogarty Visiting Fellow at the NIH and as an expert consultant for both NIH and the World Health Organization, could you share insights into collaborative efforts and global initiatives that are essential in addressing challenges posed by coronavirus on an international scale?
Well, the collaboration between labs is absolutely essential because some laboratories are expert in one area, but not in another one that is as essential as the first one. As an example, we have been collaborating once I came back to Spain from NIH. We have been collaborating with another 3 North American laboratories, one from Chicago and 2 from Iowa. NIH supported us. That was very important. But our example is very simple. We were very good experts in the engineering we call reverse genetics of coronaviruses. We were the first able to make a, let's say, synthetic coronavirus and being able to manipulate all the genes of the virus. And that was a very important tool.
That is impressive.
But we did not have the animal model. Today, to work with dangerous viruses that kill people, you have to work under very safe conditions. This is very expensive. And in general, we do all this work with mice. Why? Because mice are very small. You can keep it under safety conditions. But there was no mouse that was susceptible to human coronaviruses. And that has to be made artificially, making transgenic mice that carry the receptor for the human virus. And this important work was performed in Iowa by scientists like Stanley Perman and Paul McRae. They made models. They made little mice that look like humans in the sense that they were susceptible to the virus. So the collaboration between both laboratories was essential.
I see.
We made all the virus mutants. That was very important. We have a long-term expertise. We discovered how to do that already in year 2000. But we couldn't make experiments in animals. And they developed the first animal system susceptible to all these important coronaviruses. So the combination, the complementation between different labs is absolutely essential. And this is what we have been doing for the last almost 20 years, collaborating 3 labs from the United States, our lab far away in Spain, but this collaboration was essential.
I see. Sounds very interesting. My very last question is, as an attendee to the Viruses Conference, could you please share with us some of your impressions of this year's meeting and your thoughts on the evolution of this conference over the years?
Well, this meeting, for the moment, we have been, to now, we started at 9 o'clock, so we have many sessions, and the quality is excellent. If I want to mention an example, the first speaker was Charlie Rice. Charlie Rice is a Nobel Prize, but more than that, it has been a top virologist for many years, working with hepatitis C. This is a very difficult problem. They found, in collaboration with other teams, the solution for hepatitis C. Most of the people now can be cured, not 100%, but most of them. And it's easy to talk about Charlie Rice because he's top quality, but other presentations have been excellent. I have seen in the audience many foreigners This is very important, coming from the States or from anywhere in the world. And so it's what we call a rich meeting. There is a lot of things to learn here. I am very glad I was invited, and I hope we will get to the end with this level that we had today.
Absolutely. Thank you so much. Thank you so
You're
much for
welcome.
the interview, for answering the question. It's a pleasure. It's an honor.
Don't forget, if you have a photo with hair, if I don't have hair, don't send it to me.
Got it. No worries. Thank you.
Okay, you're welcome.